So the next company is Alzecure and Martin Jönsson. Please do your first presentation.
Thank you so much. Hello, my name is Martin Jönsson. I'm the CEO of Alzecure Pharma. Alzecure Pharma is a company who are active in Alzheimer's and pain. We are a spin-off from AstraZeneca. AstraZeneca left the space many years ago, and then Alzecure was founded by researchers from the company. We have 3 different platforms. 2 of those are in Alzheimer's. We have Alzstatin, which is a preventive disease-modifying treatment against Alzheimer's disease. And then we have new Restore, which is a treatment against cognitive disorders to help individuals improve memory and learning capabilities. And then we also have pain projects. Last week we came out with positive results on a phase 2 with ACD440 in neuropathic pain. And neuropathic pain is the largest individual pain market, and we are therefore very happy with what we have achieved. And this is with the first-in-class molecule. So things are looking good for Alzecure. So thank you.
Okay. Thank you, Martin.
Thank you.
Sit down. And my first question was a bit about ACD440,
Yes.
your pain project, and the initial phase 2 data.
Yeah.
Can you maybe just summarize the results and what they mean?
Yeah, so the results are confirming the results we got in our 1B study. There we ran a study in healthy volunteers, and we saw that we could halven the pain experience. And then we took this in to patients and did a placebo-controlled crossover study. And we got more or less the same results when it comes to pain relief. At the same time, we also saw that we didn't have any safety problems or tolerability problems. So with these data, those data are now supporting us to go on in further development of the project.
And what is the next step? I assume that you will present the data more in detail.
Absolutely. So we have accepted we have a presentation at EFIC in September. We are looking at maybe coming out with results even before that. And now next week we are going to Bio International in Boston to meet with companies and present follow-up discussions we have had before on this project. And we have a CDA with several different companies, so we look forward to talk more about both the results and also the path forward. And we have developed full development programs all the way up to phase 3. In this area, when it comes to neuropathic pain, only 1 out of 8 patients are actually experiencing adequate pain relief, so there's a huge unmet medical need. In the US, more than 50% get an opioid during their patient journey. So therefore, there is also a big interest for the society to really get good new pain medication. And what's so good with the formulation we are having, it's a topical. So therefore, we see it that we will be able, hopefully, to add to the total market, which is already there. And the market now is more than $11 billion. So to combine centrally engaging therapies with our topical gel, we think will be very attractive. So we feel very comfortable.
And you mentioned it's a very significant market,
Yes.
But how broad could you use it? Should you use it was a subset of patients, or could it be used for anyone, especially given it's a topical gel? Is there maybe some patient populations that are more relevant?
Yeah, absolutely. So if you look at neuropathic pain, about 7 to 8% of the adult population are suffering from neuropathic pain. And then about 65-70% of these are having peripheral neuropathic pain, and they have upregulated VR1 receptors, which we are targeting. And that is the patient population that we see as, how should I say, most relevant.
And how large would that be as a fraction of the total, roughly? Like, is it a significant portion at least?
It's a very significant portion. I can say that Lidoderm, they reached their peak sales in 2012, and they had a sales only in the US of 1.4 billion US dollars. And that's a patch, and the Lidoderm has a lot of side effects, and you can only use it for 12 hours. And here we have a gel which should be much easier to administrate and everything. So yeah.
And the next question then is about, we don't know too much about the data, but you have presented positive phase 2A data.
Yeah.
This was a short trial, a mechanistic trial almost. How much can this inform, like, let's say a phase 2B trial? How replicable are they in a larger context?
First of all, I can say that we did a short trial, and then of course a double crossover design, and that we got these results all on a short trial makes it actually even more impressive, because very many of the other medications need up to 4 weeks or longer to actually show effect. So that we already so quickly actually are decreasing or showing pain relief is actually quite substantial. Because what we are thinking and what we look forward to is that when we treat even for longer time periods, we can then potentially get even better effects.
And is it the same endpoint in a phase 2B, or because this was induced pain, and how would it be in a more of a natural setting in a phase 2B, or how does it compare?
It will be more, of course, in a natural setting and everything. Absolutely. Yeah. And then for us as well, we see our goal is primary to go into, how should I say, chronic pain. But there are also opportunities to actually go in acute pain to quicker come to the market. And then also to look for smaller indication where you can get a fast track and actually then do a 2B, and based on those results maybe get an approval, and then you do a phase 3 as a confirmatory. So therefore we will look at this, and our model is of course that is to out-license our projects. We have several, and to out-license them now in phase 2 or something like that. So therefore, it's also for us very much about the dialogue with potential partners or companies who want to license the product.
But as you mentioned, a smaller indication where you could get approval after a phase 2B or one trial, would that mean like an orphan indication, or?
No, there are actually indications that we have identified which are much bigger than orphan, and due to different reasons I don't want to mention the indication, but of course we could go for an orphan as well, but like this is bigger.
You can still go a shorter route but still target a larger indication.
Yes.
Okay. Yes, so maybe we can move on to Alzecure.
Yes.
Which is of course very topical with all the buzz in the Alzheimer's space. Can you maybe first off maybe just compare Alzecure to these antibodies?
Yes.
And the advantages?
Absolutely. So the antibodies are used in patients which are already diagnosed with Alzheimer, mild Alzheimer, and there the brains of the patients are broken down to 20-30%. And what the antibodies are doing, they are cleaning up the brain, and then they are halting, or they are slowing down the disease progression. With Alzecure, we see the opportunity. Our primary what we really want to achieve long term is to come with a preventive treatment which reduces the production of the protein which builds up to the plaque, and that's A beta 42. So to go into patients who are at, or individuals who are at risk, which we are identifying based on blood test, looking at A beta 42, and then combining with genetics. So you are looking for groups who are at risk. So to combining them and then identifying the patient there. But we also see the opportunity to actually potentially combine it with antibodies, or that it should be used as a follow-up, a maintenance treatment after you have been treated with an antibody. And we have a small molecule, developing it into oral medication so you can take it at home. So that's a big difference. And then also a small molecule has a higher probability of going over the blood-brain barrier. So we see Alzstatin as a very, very attractive compound, but also complementary to that antibody. So we think it's great that the antibodies are here now or are coming in, and then, but they need other.
And given that they probably at least will become the new standard of care in some ways in Alzheimer's, how dependent would an Alzstatin clinical trial be on the standard of care? Would you need to have them in some sort of control arm, or would it be totally independent of these antibodies in a clinical program?
I think it would be independent. I think so.
And when I think of Alzstatin and properties to complement the antibodies, you mentioned it's supposed to be used very early. What type of characteristics will be the most important for it to become a good complement or alternative?
First of all, it can actually be certain patient groups which actually can't be treated by antibodies, since we have seen that the antibodies aren't for everybody because of the side effects and so on. But otherwise, I mean, it is about making sure that you halt or push down the production of the amyloid beta, which is hurting the brain. And you know, Eli Lilly did, in their initial concept, they wanted to combine BACE inhibitor, which is a similar project, with their antibodies and have them combined.
And where is the status of the Alzstatin project now? What's the key upcoming goals?
Yes. So what we, it has taken several years to really have had the amyloid hypothesis confirmed. And this was confirmed in the fall. And therefore, the interest in Alzstatin has been there, but it's really boosted now with the data from lecanemab, donanemab, and so on. So what we have done now is actually we have chosen a new molecule, a new CD. And why do we do that? Both because we can choose a new CD which has better potential properties, but also to have a molecule which has a long patent time. And therefore, we chose a new molecule in January. And this molecule we already now have in safety testing and building towards to drive it through the safety testing, and then hopefully it comes out good, and then into man and do a phase 1. And what's fascinating with Alzstatin is that we believe and expect that we already in a phase 1 actually can get proof of concept, of course, but also that we in this can show efficacy data, because we should be able to see in these patients that we can lower the A beta 42 production. And we think this will be a huge value driver. And that kind of trial is not very expensive. So
like a biomarker trial then?
Oh yeah, it will be a biomarker trial. So we wouldn't be able to do cognitive tests, or so that wouldn't make sense there.
Yeah. And if we move on to ACD 856, can you maybe just where you presented interesting data here, and what is next step?
Last fall, or in the fall, we came with positive phase 1 data on NeuroRestore ACD856. We showed safety, tolerability, and also target engagement, that we are activating centers in the brain which are central for both cognition but also for depression. And then in parallel, we have done trials looking at other properties of this compound, because based on the system, when you affect BDNF and NGF, which are central for learning capability and memory capabilities, you should, based on the biology, also be able to affect brain health. And in our trials now, we actually have several data which we have done over and over to actually feel more safe, showing that we are improving neuron outgrowth, we are improving mitochondrial activity and neuroprotection, etc. So based on this, it looks like this NeuroRestore and ACD856 have potential disease-modifying abilities. And this is also, to some extent, validated by the concept of ASI. ASI also have track PAM, or track A PAM, which is focusing on NGF. And they presented phase 1 data also last summer. And based on what they saw in their trial, they actually went out and said that their compound has disease-modifying abilities. And with the NeuroRestore, we are affecting both track A and track B. So we really like their data.
And I think maybe a short final question.
Yep.
So the next step, is that a phase 2A trial then, or and would that mainly focus on the symptomatic treatments, or how would you implement this potential disease modification?
That's a great question. Initially, we will go for the symptomatic, improving learning and memory capability, and that is really what the patients really want. But at the same time, we are now looking at also how can we potentially do a disease modifying, but that is much more challenging. But we are working on trials where we should approach this. But to really make sure that we can have an approval based on this, we need to have discussion with the FDA. And as we have communicated before, we are preparing for a pre-IND meeting with NeuroRestore, because that's very central.
Yes, thank you, Martin.
Thank you, Fredrik. Thank you.